Like a message in a bottle, we have within us a symbiotic population of life forms that directly contribute to the functional quality of our lives. A human being emerges into the world based upon the blueprint for our biological creation called DNA. Every cell nucleus contains DNA in predominantly identical strands called chromosomes. But our DNA is not alone. Our gut harbours a vast diversity of DNA belonging to living things that we collectively describe as the microbiome.
Unbeknownst to many people, the majority of the cells in our body are actually microorganisms. So we have more bacteria in our body than we have human cells.
What we're talking about is the trillions of bacteria, fungi, archaea, nematodes that reside all along your gastrointestinal tract.
Caitlin Hall is a researcher at QIMR Berghofer Medical Research Institute in Brisbane, Australia. Elena Pribble is a senior scientist at Microba, a world-leading microbial genomics research and testing facility, also in Brisbane, Australia.
Those bacteria are actually playing a very key role in our bodies. And what we term the microbiome is actually the community of microorganisms that's living within our body and on our body. We also have microbes that live on our skin. But it really wasn't until the advent of advanced DNA sequencing techniques that we really started to realize what a great role these microorganisms are playing in our health and how widespread they were throughout the human body. We've identified that all of those microorganisms that live in our gut, they're all producing thousands of different substances. And those substances are actually able to interact with our human cells. They're able to activate receptors. They're able to influence the activation of different metabolic pathways in our body. They can even influence gene transcription. And so we're seeing that these substances are actually now influencing most of the bodily systems. They're modulating our immune system, they're modulating our metabolic system, and they're even modulating our nervous system.
Over the past 10 to 20 years, what we're realizing is that the health, the diversity, and the different microbial communities are really important in terms of how we experience emotions like stress, anxiety, and even depression.
And so this community of living things within us is not just interacting with digestive and intestinal activity. It influences things all over the body, including the brain. It is an extraordinary thing to discover that our emotions are affected by what is happening in our gut.
I think it's even beyond scientific research. The relationship between the gut and the brain is deeply embedded in our language and the way we communicate with each other. We say we have a gut feeling when we make decisions based on intuition. We're gutted when we're upset or disappointed, or we have butterflies in our stomach when we're nervous or anxious. So I think the scientific research is now beginning to catch up with what we've always really intuitively felt or known.
And this is a very hot topic right now for research. And so just as a bit of background, we know that the gut and the brain are connected via a superhighway we call the vagal nerve. We also know that our gut is highly innervated by the enteric nervous system. What we didn't know previously was that bidirectional communication that's occurring is actually being influenced by some of those substances that our gut bacteria are producing. So we know that 90% of the serotonin in our body is produced in our gut by certain cells called enterochromaffin cells. And we know that some of those bacterial substances, especially ones called short-chain fatty acids, can actually stimulate our enterochromaffin cells to produce serotonin. We've also know that gut bacteria can produce a lot of other neurotransmitters directly, like GABA. There's also laboratory-based evidence that gut bacteria may also be producing dopamine. However, we haven't validated the chemical pathways for that yet in bacteria.
This research is going on in laboratories all over the world and is changing our perception both medically and psychologically about how we can resolve and hopefully prevent physical and mental problems.
Our next steps in our research is to try and understand whether different intrasubject variability in terms of those serotonin and dopamine producing bacterial communities, whether that plays an effect in terms of the signaling in the brain, and if we can change the microbiome, how does that change the interaction between those brain regions? I think what this research highlights is that we do need to communicate with dietitians in the field and those that have been specifically trained to understand what to look for in the microbiome, but also what to ask in terms of what symptoms their patients are experiencing, and trying to communicate with other health professionals who know a lot about this field and the emerging research.
It wasn't too difficult to find a skillful practitioner who deals with people's gut issues on a day-to-day basis. Narelle Henschel is a practicing naturopath, herbalist and nutritionalist in Brisbane, Australia.
Sort of, I guess, a tenet of naturopathy is treat the gut. That's sort of when we're training, that's what we're taught, treat the gut, and all disease begins in the gut. I think Hippocrates was a Hippocrates saying. As a naturopath, a lot of what we do is looking at diet and lifestyle, so I'm always going to be assessing gut health and looking at what people are eating, how they're absorbing it, how they're assimilating it, and trying to improve that just as a general health thing anyway. Some people will prefer to do testing from the get-go. I sort of tend to look at the case history, the diet diary, making some changes, and then see what else we might need to do with tests. And that can be anything from standard blood tests that people would get from their doctors to look at, certain minerals and things, and then going into functional tests that look at the gut health, the enzyme secretion, the microbiome and the makeup of that.
If a naturopath had a patient that they were concerned may have a condition that's being influenced by their gut microbiome, and they want to check to see how the gut microbiome is functioning, they would take a test from us, which is basically just a swab from a piece of soiled toilet paper. Then we would take that sample and analyze it in our lab, and we'd provide a profile that's very comprehensive, listing all of the different bacterial species, as well as some of the key functions that we know are linked to health that those bacterial species are able to do. That might help guide a practitioner into understanding that maybe this person needs to start adjusting some of their lifestyle factors or their diet to improve their gut microbiome to make those values get lower, because that might be putting them at risk for inflammatory diseases or metabolic diseases.
So it's what we do with our body that can make an enormous difference. It's what we eat.
Fibre is something that is often lacking in, and we've often replaced fibre by, these, almost these supplements of fibre, or the cereal, the brown, grey cereal, and people think that that's fibre, but it's really, your fruits, your plant foods, your fruits, your vegetables, your whole grains, legumes, and a lot of those aren't in people's diets in enough quantities, and diversity as well is what we're learning more and more, that we need to eat a diversity of foods, not just 5 foods. We need to actually eat, a lot of the research is coming out, at least 30 different plant foods a week, and sometimes simply by just getting them to increase the fibre component, i.e. the plants, and the diversity, you can actually get really big shifts. And I think that's just an amazingly powerful thing. It's a supplement-free, drug-free, they've got to eat anyway, so why not eat things that are going to have all these knock-on effects across all kinds of health issues. Sometimes it even surprises me. I mean, it shouldn't, but sometimes it even surprises me.
And there are many other surprising discoveries being made possible by the advances of the analysing techniques at Microba.
Yeah, so here at Microba, we really mastered the technique for measuring the gut microbiome with precision. So our co-founders are two of the pioneers in developing really advanced sequencing techniques to be able to allow us to look at the gut microbiome in unprecedented detail. And so we've really been able to build upon their early discoveries under their supervision to put together an analysis platform where we can now actually identify between 80 to 90% of the DNA that's found in a stool sample. We finally have the technology to be able to really interrogate all these different bacterial species in our gut, understand what they're doing, and then develop new diagnostics and therapies as a result.
In practice and research, this unprecedented detail in analysis of the gut microbiome is opening doors and providing hope for those people dealing with very serious physiological and neurobiological diseases. Richard Gordon is the group leader in clinical neuroscience at the University of Queensland's Centre for Clinical Research. His team is now investigating the role of the immune system and the gut microbiome in the progression of Parkinson's disease, and perhaps even as its cause.
So the microbiome was ignored. So the first clinical description of Parkinson's was over 200 years ago in an essay by James Parkinson, after whom the disease was named. So ironically, he speculated in that essay over 200 years ago that in the 8 people that he sort of described as a shaking palsy for Parkinson's, that he speculated towards the end of his sort of classic essay that Parkinson's could be a disorder of the bowels, as they called it back in the day. So, sort of alluding to the gut-brain axis and sort of the view that all diseases could start in the gut. So, but what we're finding now over the last decade is that there are very significant, very specific changes in the gut microbiome and the gut bacteria of people with Parkinson's.
Parkinson's disease is described as a loss of dopamine in the subcortical striatal region of the brain, largely because of damage to the substantia nigra, which disrupts what is known as the nigrostriatal pathway. Parkinson's disease has been thought of predominantly as a neurodegenerative disorder, but Richard Gordon is contesting that perspective.
When a person goes to get diagnosed with Parkinson's in the clinic, right, it's typically when they have the first motor symptoms or the tremors and slowness of movement or difficulty initiating movement. So at that point, they've already lost 80% of the dopamine-producing cells in the brain, which is what triggers the symptoms. They seem to have disorders of the gastrointestinal system, so symptoms like constipation, in what we call the prodromal phase. So the prodromal phase is what comes before you have your formal disease diagnosis. So in the prodromal phase, one of the key symptoms that's common in most people with Parkinson's is constipation.
Constipation can be an early indicator that can be seen years or even decades before damage is seen in the brain.
So our research, as historically, we've focused almost entirely on the brain and the immune system in the brain and inflammation. So when we started looking at the microbiome for Parkinson's, we've found signals that could explain depression, for example, which is one of the earliest symptoms of people with Parkinson's. So that's a known motor symptom in Parkinson's. So because Parkinson's is a movement disorder, there's often non-motor symptoms that appear earlier, similar to constipation. So some of the major non-motor symptoms are constipation, sleep disorders, and depression.
Finding that indicators of these early non-motor symptoms can be found in the gut microbiome could change the way Parkinson's disease is predicted and treated. Breakthroughs in research often come with the discovery of a single molecule or single microbial interaction.
So our hope with our microbiome research is to be able to sort of understand the early signals in the microbiome that could contribute to Parkinson's disease, and either use that as a way to sort of diagnose people with Parkinson's maybe 10 years or 5 years before they lose more neurons in their brain. And we've also found evidence for increased production of trimethylamines. So trimethylamines are microbiome metabolites that are produced after they break down products found in red meat and eggs, for example, so choline-rich metabolites. So we've just got funding from the Michael J. Fox Foundation to study how trimethylamine, which is elevated in people with Parkinson's, how that could either contribute to the disease or even initiate sort of some of the pathological features that we see in people with Parkinson's. What we're trying to do now that we understand the harmful metabolic pathways, we can design drugs that could target specifically just the pathways that produce the harmful metabolites in the bacteria without actually killing the bacteria like you would do with an antibiotic. So it's a very targeted approach.
Designing these targeted drugs has required innovative techniques that are only possible with the recent developments of computer technology and molecular science. Shannon Jewell is a PhD candidate whose background is in organic synthesis.
I'm primarily focused on the drug discovery component. So we're currently working on generating disease-modifying drugs for the treatment of Parkinson's disease. So we are currently working to generate, with the use of techniques like AI and other things like that, we are developing new drugs in order to target those receptors specifically. So we have a med student currently working with us he is our main AI guy. So he's generating hundreds and hundreds of molecules using that technology. He will input our protein target along with known agonists for this protein target, so natural agonists for it. And then what the software does is actually generate literally hundreds and hundreds of molecules that have similar structures and high binding affinity for this molecule. And then what we can do is we can take all of those structures that this AI has developed for us and pick and choose which ones are chemically stable and which ones are chemically ridiculous, right? Because it does generate a fair few of those as well. So we can pick and choose for things like if we want it to cross the blood-brain barrier, we can select for something like that. Or if we want it to be targeting the gut, we can select for that based on the structure of the molecules that they generate, yeah.
So our ultimate hope is to be able to develop treatments where we can slow or stop the progression of Parkinson's. So even if we can slow the disease by say 10% or 15%, because there's ways to manage it, that means that we can dramatically improve a person's quality of life and sort of prevent this debilitating progression that happens from year to year.
It's amazing because it's completely untapped, right? Nobody's really thinking to target the gut for Parkinson's disease, or it's a very new idea. So there's so much potential there, and it's really incredible.
The presence of mood disorders, or what are also called affective disorders like depression and anxiety, are not just related to Parkinson's disease. Many people struggle with mood disorders and find that they are being treated as though these moods are all in the head. Investigations of the gut microbiome in people with mood disorders are revealing breakthrough insights into the importance and impact of the gut-brain connection, a communication system that occurs in both directions.
We know that the gut microbiome, like in people that have major depressive disorder, is different from the gut microbiome of people that are healthy. But now we're going to actually be able to start looking at, okay, so which bacteria are different in people that are healthy compared to people with depressive disorder? Let's try to get a consistent signal. And beyond just looking at bacterial species, let's also look at what are the functions that are different.
Now that we've established a relationship between the diversity, the composition, and the function of the human gut microbiome, both in health and also in disease states like inflammatory bowel disease, how can we now change the microbiome or look at altering the interaction between those microbial communities that can facilitate and potentially alleviate those symptoms of mental health, including anxiety and depression. The Western diet may be contributing to some of the changes in the microbiome that we've been seeing over time. I think a pretty popular opinion at the moment is that by changing the fiber and by changing the quality of diet that we're consuming, so fruits, vegetables, legumes, nuts, seeds, can only help the microbiome and alongside that help mental health outcomes as well.
Probably young women in their like 20s or late teens and been diagnosed with IBS, and then there's this anxiety around it, and maybe they've been put on hormonal contraception as well, which often feeds into, can feed into leaky gut and just general dysbiosis when you've been on it a long period of time. St. John's wort, which a lot of people, they kind of label it as a herbal antidepressant, which is not quite correct because herbs have like hundreds of phytochemicals in them, so they're not targeting. But how old school, if you read old school herbal texts, look at St. John's wort, is that it actually balances the enteric nervous system. So it's working from the gut to affect the brain. So it is acting sort of an antidepressant, but not in the way people are thinking, like a serotonin, the SSRIs or things like that.
So we can expand that old saying of gut feeling further than just intuition. The condition of our gut and the diversity and health of our microbia are all part of our emotional experience. There certainly are things going on in the head, but some of these things are in relation to what is going on in our gut. This means that we have more ways to facilitate recovery and develop ongoing resilience.
But we do see, like in mouse models, that if you feed mice prebiotic fiber or short-chain fatty acids, that helps alleviate stress and anxiety in mice. And there was actually a single human study that was done in type 2 diabetic patients, and they were fed a prebiotic fiber over the course of several weeks, and they did show that over the course of that experiment that scores on anxiety and depression tests were actually reduced in the group that was fed the prebiotic fiber compared to the group that just was given a placebo.
So my research is looking at trying to bring some of that really fantastic preclinical work, so studies in mice and rodents, and trying to bring that to the more complex human situation. So sometimes studies in mice and animals don't necessarily replicate to humans because there's been really controlled housing, diet, sleeping conditions, and humans are completely different in the way that we interact with our world. So our research is looking at neuroimaging techniques, so specifically functional magnetic resonance imaging, so fMRI, and also looking at sequencing the gut microbiome to try and understand specifically if we're feeling anxious or if we're feeling worried about something, how that translates to how our brain regions communicate with one another. So if you have antibiotics, for example, if you have some sort of disruption to the health of your microbiome, how does that look in terms of how the brain activity responds to threat or safety cues in the environment or different emotional responses? So we've done this research both in healthy individuals, so people who have a normal functioning microbiome, and we also look at the differences in the gut-brain responses in patients with inflammatory bowel disease. So inflammatory bowel disease is made up of kind of two main sub-disease groups, which is Crohn's disease and ulcerative colitis. And it provides a really compelling example of what happens when the microbiome is upset or dysregulated. So what we find in our Crohn's patients, for example, is that if they've had an inflammatory response or if they've had some sort of major disruption to their microbiome health, we see a really disrupted pattern of communication between key brain regions important for emotional regulation and responding to anxiety and threat signals in our environment. So we already know that patients with inflammatory bowel disease do have very high levels of anxiety and depression, but what we've found is that the signaling between these two key brain regions, which is the insular cortex and the medial prefrontal cortex, is increased in our IBD patients.
Research facilities like Microba, QIMR Berghofer Medical Research Institute and the University of Queensland are leading contributors to the worldwide investigation of the gut microbiota and what this will mean not only for physical but also our mental and emotional health and well-being. Practitioners like Narelle Henschel are translating the research into functional benefits for people in their community. But the researchers know that there is much more to learn. We're just beginning to understand what all the many microscopic elements are doing and how they function together.
We're also seeing gut bacteria influencing the production of hormones that communicate with our brain. So like appetite regulating hormones. Those same enterochromaffin cells can also be stimulated to produce peptide YY or glucagon-like peptide 1, which we know are very important to send messages to our brain that, we're full, we need to stop eating. And if you don't have your gut bacteria producing those beneficial short-chain fatty acids, then those important hormones aren't being produced. Your brain's not getting the signals that you need to stop eating. And so it could also be contributing not only to potentially mental disorders, but also metabolic disease.
So we're still in the very early stages of this research and really beginning to get at those more complex questions about if we can see a change in terms of the microbiome and how that impacts outcomes like stress, mood and anxiety. What can we do to change the environment in the microbiome to support mental health? And at the moment, one of the key interventions that researchers, including us, are looking at is how we can create a probiotic or a prebiotic or some sort of dietary intervention that can support the health of the microbiome and to see whether that can change how our brain regions communicate and also change those behavioral outcomes. And I think that this is a cost-effective, a low-risk intervention that we can use.
Finding these links between the quality of our gut bacteria, emotional health, easy-to-use supplements, and simply paying attention to our diet is generating interesting research and interesting discoveries.
So one thing that has been really interesting that's come out is that a lot of the Western diet type foods really promote the growth of pro-inflammatory bacteria. And these are bacteria that produce lipopolysaccharide. Now this is a key bacterial substance that's known to be able to activate immune cell receptors to trigger inflammation throughout the body. And so what we see is that people that have a Western diet typically have more bacteria that are producing this lipopolysaccharide. When you eat a high-fat diet, this lipopolysaccharide is then able to basically hitch a ride on some molecules that would typically transport fat across your cell barriers to the rest of your body. And your body just sees this lipopolysaccharide as a fat molecule, so it gets picked up as well and is thus put into your body's circulation. And anytime that molecule now comes into contact with an immune cell, it's triggering inflammation. And it's a low-grade inflammation, so it's not typically that noticeable, but over time, that builds up and starts being a major risk factor for chronic disease.
So over the last few months, we've started looking at the specific metabolites produced by the microbiome and how they could contribute to the disease. So we've identified things like the trimethylamine, which is a microbiome metabolite linked to cardiovascular disease. And we found that in Parkinson's, this metabolite can trigger inflammation, specific inflammation pathways. And it could also contribute to some of the protein misfolding that's linked to Parkinson's. So the Lewy body aggregates that are found in the gut and in the brain. And there's a bit of speculation as to how it spreads from the gut to the brain, or if it originates in the brain itself. But we found that bacterial metabolites can contribute to that protein misfolding and that seeding of those aggregates that accumulate in people with Parkinson's.
One of the most interesting aspects that I've found that's been coming out in gut microbiome research is the ability of some of these microbial substances to actually impact our blood-brain barrier. Now, I find this quite surprising because it's not typically the first thing you think of. You'd really think, these metabolites would be, signaling, neurons in our gut, and signals would be traveling that way. But it seems that some of these metabolites, they do get into our blood circulation, and they can actually go directly to our brain and influence that blood-brain barrier. It was about a year or two ago, I think, where they were looking at some cadavers of people that had died from Alzheimer's disease, and they did find what they thought was Porphyromonas gingivalis, a potential pathobiont that's found in our mouth typically, up in the brain of these patients. And so again, that was just a single study. It needs to be confirmed, but it also suggests that the weakening of that blood-brain barrier could potentially be letting in, bacteria all the way up there, which could be stimulating neuroinflammation.
Understanding the bidirectional links and processes between the gut and the brain has the potential to change our therapeutic approach to physical and mental health. With the advanced DNA sequencing now available, it is only a matter of time and a little more research.
There are very exciting links emerging between early chemical exposure in your sort of 20s, 30s, and 40s, which could decimate the microbiome, right? And then those sort of early life events or midlife events in terms of chemical exposures, while they're not antibiotics and they don't destroy the microbiome, they do have an adverse impact, which can then lead to sort of diseases like Parkinson's later in life. We are taking that approach with metabolites like trimethylamine, which we found to be elevated in people with Parkinson's. So one of the funding applications we're putting in is to be able to target the production of trimethylamine in bacteria without actually killing them with antibiotics. So you don't decimate entire population of the bacteria.
I don't think it's necessarily the impact of a single species or strain that's having these effects in the brain. What we're now beginning to understand is that it's the relationships between these microbial communities and how they interact with each other and how they engage in cross-feeding behaviours that could be really important in terms of the next steps in this research area. A very complex system, so necessarily changing one species is not going to have long-term or massive effects. What we really should be thinking about is how can we support the whole community and the diversity of microbes that exist there, and working out what that will be, I think, is going to be a very big challenge in the future. There are several challenges in the field at the moment. One of them is that humans are far more complex than mice or animals, in that it's very hard to control our diets, hard to control, the work environment or stresses that we experience.
And so by recognizing that, it's something that's very easily to change. I mean, that's the great thing about our gut microbiome, is it's very malleable. It's not something that we're set with like our genetics. It's something that's influenced primarily by diet and also by our lifestyle, so how much we exercise, how we manage our stress, medications we take. And so by making some changes in our lifestyle, it'd be quite easy to reduce the number of those bacteria producing these pro-inflammatory molecules and increase those beneficial bacteria producing those short-chain fatty acids that we know are going to help promote serotonin production and reduce inflammation.
Clearly, there is still much to learn, and there are many breakthroughs yet to come, but we can all do something positive right now. Elena Pribble leaves us with a simple recipe.
The key to all of this is really having a healthy diet, trying to go back to eating more plant-based whole foods, trying to reduce the saturated fats and the processed foods from our diet. Basically everything we've already known and that we've grown up with is being taught what is a healthy diet. We just need to follow that.